Hello guys, my name is Dr. Varun and you are watching Chlorophyll classes on YouTube. So till now for 12th class we were doing principle of inheritance topic, and in that topic we have reached to the point of interactions which are post-Mendelian interactions. In post-Mendelian interactions till now whatever we have studied, that was the allelic genetic inheritance or interactions, means within the same alleles we were working on. I hope that the allele concept is clear to everyone. If not, then let me give you an example, quick example over here. Imagine that this is the chromosomes, and on chromosomes there are what, multiple different genes present, and these homologous chromosomes are there. So on a homologous chromosomes, this is representing what? It is representing gene. Okay, and this is are the two different alleles of it. Maybe the alleles could be similar, then the condition would be homozygous. If the alleles are different, contrasting, the condition becomes heterozygous. Okay, non-allelic means what? Now this one gene, for example this A gene, and here we have another gene, this B. Okay, now they both are interacting. So now the interactions are what? Between different alleles, okay, of the different genes. So means different genes are working in this case. It can be present on the homologous chromosome or on the heterologous chromosome, means on a different chromosomes also, but they are affecting. So this is the one we are studying over here. Okay, so I'm just gonna be writing this thing super quick. So what are they? These are the interactions between genes located at different locations or different loci on same chromosome or different chromosomes. So we reach to one another steps, that we are not just confirming ourselves with what within the same alleles. Okay, we are going towards the different alleles of different genes now. So here we have multiple of them. So what are those? Number one, we're gonna be starting with the complementary genes interactions. Complementary genes. Now complementary means that if you are getting one thing, another thing is added complementary to it. Like for example, if you are going to buy a pizza and you are getting Pepsi free with it, okay, that is complementary with it. So this same thing comes over here. So means one gene along with another gene will work, something new will form. So here the example is, we're going to be doing the example of sweet pea. Okay, so in sweet pea, what happened? There is a purple color flowers that are being formed. Now to form a purple color flowers, two genes are acting together. Let's say one gene is C, other is P. So when the capital C, capital P will together present, it will give what? It will give the purple color. Or else if they are not present together, they will give what? White color flowers. Okay, so this thing we're gonna be writing up. So let's just see that. So we're gonna be writing like what is happening in detail. So detail is happening what? So basically these genes are making certain pigments or certain substrate. So for example, imagine that you have a substrate. So you are making what? A dye. Okay, so if anyone have seen that how we dye a cloth, so basically what happened, we put the cloth into a colored medium so that it got the color. Okay, same thing over here. So imagine that we have a substrate. Okay, this substrate, when it is acted by what? Gene capital C, it leads to formation of a substance called as your substrate B. Okay, so this second substrate has been formed, substrate B, I'm saying it, and on this what happened? There your gene P occur, which made a compound that is anthocyanin. Okay, which is giving what? That is giving you what? It's giving you your color. Means if only C would be present, then the color won't come. If only P would be present, not the color come. Both has to be present. Let's just see that how it will occur. So for example, if you have a two plants, okay, let's see, the first one is capital C and small p's, other is small c, capital P. In both the cases, the sweet pea plant will have white flowers. White flower. Okay, so I am just making a flower like this. Okay, so white flower is there. Now it will make what? Gametes. So capital P, small, sorry, capital C, small p, small and capital. So these are what? These are the gametes. Okay, now what will happen? There will be crossing. This fertilization leads to what? Capital, small, capital, small. So I told you that once the capital C and capital P will come, it will give us what? It will give us the color. That is what? A purple color. So this will be a purple colored flower. So let's just now when this parent, this F1 generation parent, it undergo what? It undergoes selfing. Then what will happen? That is the major thing. Before that, okay, so we're gonna be making the Punnett square out of it. Okay, so what would be the gametes? Capital C, capital P, capital C, small p, small c, capital, small, small. Okay, same here. So when you make this square completely, let's see that. Quickly make it. Okay, let's just now see in this that what is happening and what kind of flowers will come. We know that purple color flowers will come when there will be capital C and capital P at least one present. Okay, so this is here, this is here, this is here, this is here, this two, this two, this two, this also, that's it. And the white color flower would be when there is a no cap, right, when both the capitals are not present. Here both the capitals not present, both capitals not present, not present, not present, not present, not present, not present. So let's just calculate it. How many, like phenotypical ratio, phenotypic ratio. In phenotypic ratio, what are the colors? We have purple ratio white. So how many purple present? Let's see that. 1 2 3 4 5 6 7 8 9. So 9 are what? 9 are your purple, remaining 7 would be white. 1 2 3 4 5 6 7. Okay, so this is the phenotypic ratio. What about the genotypic ratio? Genotypic ratio. The genotypic ratio is not so important for us, but if you want to write it, they can ask you. In case of dihybrid, there might be there. So that would be similar over here. No change in the genotypic ratio, same like the dihybrid ratio of the Mendel. Okay, because genes are not our concern over here. Okay, only the phenotypic ratio is the major thing. But if we see if they ask you that what is the difference between the phenotypic ratio, then the phenotypic ratio normally of the Mendel was what? 9 ratio 3 ratio 3 ratio 1. Here it is not that, it is 9 ratio 7. So this becomes what? Your first interaction, that is the complementary gene interactions. Okay, now after complementary gene interactions, we're going to be going with the supplementary gene interactions. So next one is second that we're going to be studying today is supplementary gene interaction. Now in supplementary gene interactions, again the color phenomena will work. Okay, what would be the color phenomena? Now normally there is what? Substrate level working we're going to be seeing over here. In this we will be taking the example of green color in maize. Okay, so for the color certain genes are working. Let's take that there is a two genes which I'm taking, R and P. Okay, so basically what happened that, for example, you have a substrate. Okay, if on this substrate gene R works, then it leads to what? A red color green. But if on the top of that, if the gene P also works, then what happened? A purple green color comes up. So what happened? When the two genes are working together, then they will work what? Like the dominant one of it, they will give different colors. Okay, let's just take an example. This is white, no color. So let's just make a cross. So initially we have what? R, P, P, and small r, capital P, capital P. Now individually they are not able to work. Together only they can show their effects. So individually this would be what? White in color, or this will be also a white color green. So I'm making the green like this, white color. Now what will happen? These are the parents, they will make the gametes. So these would be the gametes. Okay, there will be a fertilization, and this would be the case. Now it has what? It has capital R also, capital P also, capital R also, capital P also. Means which color will come? Purple color will come. So this grain size would be what? It will be purple. So what happened? It has supplementary, means extra things present, extra effects coming up. Okay, so now let's just see that what is happening when it will cross. Selfing will occur. Selfing. So selfing would be what? It will be the F2 generation. So we're gonna be making what? Punnett square. So what would be the gamete? Capital, capital and small, small and capital, small. Okay. So now what would be the final? This is how your whole cross would be. Practice as much as possible so that there you have a better grip over it, because in your board exams these things will come. Let's just see that what will happen. So what we have to see, we have to see the white color ones, we have to see the red color ones, and we have to see the purple color ones. So red color will come when capital R will come only. Purple color will come with the capital R should also be there, capital P should be also there. Let's just first find the capital red ones only. Here both are there, so it won't. Both are there, won't. Both. So no one in the first line. So in first line all would be what? Both are there means purple. They all will be purple. This is also both are coming purple, 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 purple. These are the maximum purple ones coming. Let's see the red one. For red one, only capital R should be there. There should not be any capital P. So capital R is here, capital R is here, capital R is here. Now the rest one is what? When where the capital P is there, but alone P won't be able to do the works. Capital R is also needed. So rest all will appear what? All will appears to be this. So this becomes what? Your all the colors that will come. So here again we have to find what? Phenotype, genotype. So you don't know, you don't need it. Okay, so phenotypic ratio. So what would be the phenotypic ratio? 3 different colors will come. What are the 3 different colors? Purple will come. Okay, purple will come, red will come, and white will come. So how many purple? Let's count it. 1 2 3 4 5 6 7 8 9. So 9. Red is 3, white is 4. So this is the phenotypic ratio. See how the phenotypic ratio is changing in non-allelic inheritance. Okay, in complementary it was 9:7. Here it is 9:3:4. Okay, so this is what? Supplementary gene interactions. After supplementary gene interactions, the next interactions that we're gonna be doing is the epistasis. Okay, so what is, okay, before going to epistasis, let's do the duplicatory genes. It's much easier. Okay, before going to the epistasis. So the third one which we're gonna be doing is the duplicate genes. Duplicate genes. Now in duplicate genes, what happened? Genes which are acting like they are present on the different sites but acting similar. Okay, whether they inheritance together or separately, they will show the same effects. So basically what happened, if there are two dihybrid cross in two different genes are there, and one gene is coming in dominant, both gene is coming in dominant, or either of it coming in dominant, it will show what? One character, dominant character. If both are coming in the recessive, then only what? A different thing will come. So here the ratio finding is very easy, because what happened? Only one case is there where the recessive trait comes. Okay, so there will be 15:1 phenotype. Let's just see that what is happening. So in duplicate genes, what happened? When genes are inherited together or separately, but still shows dominant effect let's just see the example over here. Here the example is what? Klebsiella. Okay, so the example is of Klebsiella. Now what happened in capsula, their shape of the pod is what? It is two different. Okay, the capsule of the capsula has two different shapes. One is triangular, other is top shape. Top shape you might know na, like earlier students used to play, like children used to play from this top kind of a thing, like this way, and it's keep on moving. Okay, or it's do like this and keep on moving. That is top shape. So I'm making that what? Klebsiella capsule has two shapes. Okay, has two different shapes, that is triangular or top shaped. Triangular means like this, top shaped means like this. Okay, so this thing you have to remember. Now the triangular is the dominant one over the top shape. So top shape will only be coming when the recessive trait would be there. Okay, because already the two genes are working on it. Okay, so let's just see that two genes are working on formation of it. Let's see that this capital ones. Okay, it is what? It is giving a triangular shape, whereas this recessive trait is giving what? Top shape. Okay, now what would be the gametes? This would be the gametes. So these are what? Gametes, parents. Cross will occur. Okay, so this thing will come up what? Since the both of them are present, I told you that even a single capital coming or the both is coming, it will show what? It will show the triangular one, which is the dominant trait. Now we said what? We did the selfing of it. Now in selfing again, we have to make what? We have to make our Punnett square. Punnett square would be same. Okay, so let's just quickly make it. So we have capital A, capital B, capital small. These will be the gametes. Once you will practice it, you can also write it fast. Okay. Okay, let's just, I told you what? I told you that the triangular ones would be what? The triangular ones would be what? Which will have either of the one or both of the capitals. So there are both of the capitals: triangular, 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 because one is coming. So which do not have? The last one. So this will only be what? The top shape. So the phenotypic ratio that we're gonna be concerned about in each of them, the phenotypic ratio of this, that is triangular versus top shaped, is what? 15:1. So this is about the duplicate genes. After duplicate genes, the next is the epistasis. Okay, so what is epistasis? Epistasis is that something is masking the effects of other. Means what happened if one particular type of gene is present, the other's feature would be masked. So in epistasis, I told you what? It is the one gene is masking the effects of another. Genes marks or masked or hide. Mask means hide. Gene masked the effect or action of another non-allelic gene, and this term is called as epistatic gene. The gene is known as epistatic gene, and the process is known as epistasis. Now the gene whose effect is masked is called as hypostatic genes. Okay, that it is below the effects. Okay, so gene whose action is masked, known as, this Ka means known as hypostatic gene. Hypostatic means below the effect, okay, of the gene. Now it is of two types. One is dominant, other is the recessive one. Let's just first start with the, within it there is what? Recessive. Number one is recessive epistasis. Now here we're gonna be taking the example of coat color of mice. Coat color of mice. देखिए हर्ष का फोन आ रहा है, एक बार देखो. Coat color of mice. In coat color of mice, you have to remember that two genes are working, and there will be formation of three different colors. So what are the three different colors? You might have seen that there are gray color mice, there are colored mice which is brownish in color, and there is a white mice. That is, you might have watched the movie like Stuart Little. So it is white in color. How it is coming white? Because of what? Certain genes which are working. Let's see that. Now I am saying that two different genes are working. One I am taking it with C, other is with the A. Now the C and A word itself meaning a lot of it. Means what? If only capital C is present, gene present, even in the dominant, like single present, or maybe present in the double form. So if only C, capital C gene present, okay, then C means colored. So means the mice coat would be what? Colored. Okay, colored coat would be there. If only A present, A means albino. Albino means white, means two or little well produced. Okay, so if only A present means what? Albino coat color will be there. And if C and A together present, if capital C and capital A gene present together, then what will happen? A will stop the work of C. Okay, and because A is what? Recessive color, because the albino color is recessive color. White color is considered as a recessive color. Okay, it don't have a dominant feature. So here what is happening? That this, like, A is recessive, and this is what? C is dominant. Now if C and A both are present together, then what will happen? Color that would come, that would be a gouty. A gouty is what? It is a gray color. So here what happened? Here this A masked the activity of C. Masked C. So C become what? C become hypostatic gene, and that is what our concept is. The recessive means the recessive gene is masking the effect of a dominant gene. What was the recessive? A was recessive. Who masked the effect of what? C, which was dominant. I told you. So this you have to remember. If you understand this, you will know everything. If not, then let me know in the comment section. I will make a short out of it, or maybe I have already made a short out of it by the time you will watch the videos. Okay, so let's just see that what gonna happen in the cross. So in the cross, there is what? I am taking one colored mice. So the colored is what? Only I need a capital C. Okay, so it will be what? Colored mice. Colored mice coat. And for albino, we have to leave what? Small this and capital A. This is what? Albino mice. Right? Now, their gametes would be what? Capital, small, small, capital. These are what? These are the gametes. Parents, please make a habit of writing all those pointers. I'm not writing, I'm writing very less when I'm doing it, but you have to write everything in your board exams to have a practice of it. Don't just sit, just write with me. Maybe pause it and make everything and find it out. Okay, so parents, this is the gametes. There will be what? Cross that will occur. The cross leads to what? This. So this is what? Capital C, capital A, means A will act like what? A will do, it will do the recessive epistasis, means what will happen? There will be a epistatic, recessive epistatic phenomena will occur, and this all thing would be what? A gouty colored mice. आ जाओ कॉल किया था, क्या हुआ?
अरे मैंने उसको ऐसे कि लड्डू खाने हैं वो तो मैं ले आता है।
अच्छा, फिर निकी ने बता दिया?
हाँ, क्लास ले लिया वो वाली YouTube के लिए।
So now we're going to be doing what? F2 generation, which is basically what? It is selfing that we're going to be doing. So we're going to be making our Punnett square. चला लो, AC चलाने तो
वो आवाज आएगी ना तो
कर लो चल जाएगा। AC फैन की ज़्यादा आवाज़ होती है ना।
आज फिर हो गई थी एलर्जी।
हाँ बता रही थी
आज यहाँ पे हुई थी, दिख रहा है ये इतने एरिया पे। मुझे वही लग रहा है सर्दी गर्मी वाला केस।
हाँ।
क्योंकि यही हुआ था उस दिन भी, हम लोग बहुत गर्मी में थे और फिर यहाँ के एसी में आए, और ये हुई थी रात में जब हम लोग एसी में आए थे।
हाँ।
वैसे नहीं हुई थी। और आज भी यही केस हुआ, वहाँ पे पहले गर्मी थी फिर बारिश हो गई, थोड़ा ठंडा हुआ मौसम, तो जैसे ठंडा हुआ मौसम वैसी एलर्जी हो गई।
Okay, coming back to the normal, that is, this is our gametes that gonna be produced. So let's just completely fill it up. Now a lot of you might see that why shouldn't we write it like this? Because beta, when the genes are written, they will be written in a pair form. This don't look like a pair form. Okay, so this lot of students do the mistake that we will write like this, but that's the wrong way of writing it. We write it in the paired form. Okay, so pairing would be like this only. So this is your Punnett square. This might seems little tougher than the rest, but this is, if you practice it more, it will come. Let's just make it colored mice. Colored mice means what? There should be only what? There should be capital C. Okay, not other anything. Okay, the A will be smaller only. So if you see that here it is both, it is. So this will be colored. Here both, this would be there. Here both, here it is not there, here. So at these three places only the colored mice would be there. Where would be the albino ones? Albino ones where A is the capital one, C is not present in the capitals. So this will give albino. Okay, albino means because there won't be any color. Okay, this will give albino. Rest everywhere capital C is coming. Once the capital C and they both are coming, then the color will become what? It will becomes gray. Okay, so this would be what? Gray means agouti. This one. So this is the table look like. So if I have to write the phenotype of it, because that is only we are more concerned about, phenotypic ratio. So there is what? There is colored. Okay, the more one is agouti. So agouti agouti, ratio colored, ratio white, or albino. Albino. So we have got 4 albino, 3 this, and this is 1, 2, 3, 4, 5, 6, 7, 8, 9. 9. So this becomes the recessive epistasic ratio. Okay, I hope this thing is understood to you. Remember this fact, you would be able to do this. Okay, so these are the recessive epistas. The next one that we're gonna be today is, this is the last one, that is the dominant epistas, which is the another version of what epistats. So this is what dominant epistasis. So in the dominant epistasis, what happens? That it occurs in case of what? It occurs in the summer squash, which is called as cucurbita pepo. So example is what we are taking? We are taking the fruit color of summer squash. That is what? Cucurbita pepo, scientific name. Now what happened in them? In them there are 3 different types of colors that is there. There is a white, green and yellow. Okay, over here what are the colors? How it will comes? Now the yellow color, white color is dominant over all the colors. Yellow is dominant over the green. Okay, so what happened? White is dominant over what? Over yellow, and yellow is dominant over green. So white is dominant over this and yellow is dominant over this. Now how the colors will come? So white we're gonna be writing it with the W, yellow we're gonna be writing with this, and if nothing is coming up, means all are recessive, then green. That is only one case, we know that. Okay, so let's just see that how what is happening. So if we have a 2 plants, okay, one is white, so white would be what? Like this. Here what is happening? Even the capital Y is W is dominant. So if the capital Y is even present over here, if you see, still this will what? Capital W is present. So W will show what? It will show the white color because it is dominant, no matter it is present in 2 number or 1 number. Okay, so this thing will get what? It becomes what? Hypostatic gene in this case. Okay, another plant that I am taking is what? This small. This is what? It do not have either of capitals, means it will show what? Green color. So this is the green color one, fruit plant, and this is the white color one. Okay, now what will happen? These will be, they will make the gametes. So these are the parents and they will make the gametes. So capital, capital. Here would be small, small. Okay, so these will be the gametes. Okay, it seems okay. Now the F1 generation that will be produced. The F1 generation would be what? It will be mix of this, that is capital, small, capital, small. Okay, now we are doing what? We are doing the selfing of it. Again, let's make a Punnett square. So F2 generation would be like, so what would be the gametes? Same thing over here again, capital. So other would be the similar gametes. So capital, Okay, let's just see that what gonna happen. So what would be the white one where the capital W is present, irrespective of what is present over here. So if I'm writing that capital W coming means white, if only capital Y is there, then it will be yellow, and no capital this and this will give green. Okay, this is the fact you have to remember. So the white would be what? Here capital W, capital W, capital W, capital, 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 capital. So these would be what? Phenotypic ratio of white. So phenotypic ratio. So what is white? How many yellow and how many green? So white are these much. Let's just count it. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. So 12s are what? White. Then comes the yellow one. The yellow one means the capital Y should be there, no capital W should be there. This. So 3 this. And green is what? Green would be only this one because it neither has a capital W, capital Y. So 1. This becomes the phenotypic ratio. With this dominant epistasis over. Okay, so I hope that this thing is understood to you. Well, for today's video, this is the maximum we are doing with it. So what we have completed different type of non-allelic gene interactions. Means we go beyond the one single gene, we go to the other genes which is showing the effects in certain plants. We have studied what? We have studied about the complementary genes, we have studied about the supplementary genes, we have studied about the duplicate genes and epistasis. In epistasis, we studied two, that is dominant and the recessive ones. There are other also presence like lethal genes interactions, there is inhibitory genes interactions, there is collaborative gene interaction, additive gene interactions, which we're gonna be studying later on if needed. For now, it is not needed for you. This much is the most important part which is needed to you. Well, in this video, we have completed a lot of parts. In next video, we would be completing what we're gonna be seeing about the polygenic inheritance, that is quantitative inheritance. We're gonna be seeing about the pleiotropy. We're gonna be seeing about the different types of crosses like cytoplasmic inheritance. We're gonna be seeing about the reciprocal cross, maternal inheritance. We're gonna be seeing test cross, back cross, which is also one of the very important topic for the boards as well as for the NEET examinations. That's gonna be taking place tomorrow. If you like this video, give it a like, share it with your friends, subscribe if you haven't yet subscribed with your friends. Do let me know in the comment section what are the things you understand and what better can be done on this video. Well, that's all for this video. Thank you.